HESI A2 Biology Domains and Kingdoms. Question 1. Genetically similar cells are maintained under standardized culture conditions. During protein synthesis in a laboratory cell line, which molecule carries a specific amino acid and pairs its anticodon with an mRNA codon? Option A, messenger RNA only. Option B, DNA polymerase. Option C, transfer RNA. Option D, phospholipid. The correct answer is option C, transfer RNA. An adapter molecule links the nucleotide code to the amino-acid sequence. One region binds a particular amino acid, while another contains a complementary anticodon. This matching allows the ribosome to add the correct subunit to the growing chain. The evidence from a laboratory cell line applies this principle in a concrete biological setting. Question 2. Genetically similar cells are maintained under standardized culture conditions. A single nucleotide is replaced in a gene from a laboratory cell line, but the encoded amino acid remains unchanged. Which feature of the genetic code explains this result? Option A, codons are made of amino acids. Option B, more than one codon can specify the same amino acid. Option C, proteins are copied directly from DNA without RNA. Option D, every mutation removes a chromosome. The correct answer is option B, more than one codon can specify the same amino acid. The code is redundant, meaning several triplets can correspond to the same amino acid. A base substitution may therefore change a codon without changing the polypeptide sequence. Such a change can be silent even though the DNA sequence has been altered. The evidence from a laboratory cell line applies this principle in a concrete biological setting. Question 3. Genetically similar cells are maintained under standardized culture conditions. During a laboratory cell line, damaged organelles accumulate because acidic digestive vesicles cannot break them down. Which immediate cellular consequence is most likely? Option A, the plasma membrane becomes a chromosome. Option B, DNA replication accelerates in every cell. Option C, photosynthesis begins in animal cells. Option D, worn components and macromolecules build up in the cytoplasm. The correct answer is option D, worn components and macromolecules build up in the cytoplasm. Intracellular recycling depends on vesicles containing hydrolytic enzymes. When those enzymes or compartments fail, cellular debris is not efficiently degraded and reused. Accumulation can interfere with normal metabolism and cell survival. The evidence from a laboratory cell line applies this principle in a concrete biological setting. Stressed about your HESI A2 exam? We can take HESI A2 exam for you remotely and undetected. We always guarantee a pass and payments only after results. To send us an email, scan the code or tap the link in the description and let's get it done. Question 4. Genetically similar cells are maintained under standardized culture conditions. Ribosomes in a laboratory cell line read a messenger RNA sequence in groups of 3 nucleotides. What does each group usually specify? Option A, a phospholipid bilayer. Option B, a new nucleus. Option C, an amino acid or a start or stop signal. Option D, a complete chromosome. The correct answer is option C, an amino acid or a start or stop signal. The genetic code is organized into 3 base units. Transfer RNAs match these units and deliver the corresponding amino acids during polypeptide assembly. Certain units instead signal initiation or termination of translation. The evidence from a laboratory cell line applies this principle in a concrete biological setting. Question 5. Genetically similar cells are maintained under standardized culture conditions. A population survey in a laboratory cell line uses only 2 organisms, while a second survey uses 50 randomly selected organisms. Why is the larger sample generally preferable? Option A, it removes all need for random selection. Option B, it proves causation without an experiment. Option C, it guarantees that every measurement is identical. Option D, it is more likely to represent population variation and is less affected by one unusual individual. The correct answer is option D, it is more likely to represent population variation and is less affected by one unusual individual. Larger samples usually capture a broader range of natural variation. Individual outliers have less influence on the overall estimate when more observations are included. Sampling method still matters because a large biased sample can remain unrepresentative. The evidence from a laboratory cell line applies this principle in a concrete biological setting. Question 6. Genetically similar cells are maintained under standardized culture conditions. A cell in a laboratory cell line loses internal protein fibers that maintain shape and move vesicles. Which cellular system has been disrupted? Option A, cytoskeleton. Option B, nucleolus. Option C, chromatin. Option D, cell wall. The correct answer is option A, cytoskeleton. Networks of protein filaments support cell shape, position organelles, and provide tracks for intracellular transport. They also participate in cell movement and division. A rigid extracellular wall cannot substitute for these dynamic internal functions. The evidence from a laboratory cell line applies this principle in a concrete biological setting. Question 7. Genetically similar cells are maintained under standardized culture conditions. For a heterozygous monohybrid cross in a laboratory cell line with complete dominance, which phenotype ratio is expected? Option A, all recessive. Option B, 1 dominant, 1 recessive. Option C, 3 dominant, 1 recessive. Option D, 1 dominant, 3 recessive. The correct answer is option C, 3 dominant, 1 recessive. 3 of the 4 genotype combinations contain at least 1 dominant allele. Those 3 express the dominant phenotype, while the homozygous recessive combination produces the alternate trait. Phenotype grouping therefore differs from the 1- to -2- to -1 genotype ratio. The evidence from a laboratory cell line applies this principle in a concrete biological setting. Question 8. Genetically similar cells are maintained under standardized culture conditions. In a plant population from a laboratory cell line, red and white alleles show incomplete dominance, and heterozygotes are pink. Which cross produces red, pink, and white offspring in a 1:2:1 ratio? Option A, white times white. Option B, red times white only. Option C, red times red. Option D, pink times pink. The correct answer is option D, pink times pink. Each intermediate -colored parent carries 1 red allele and 1 white allele. Their gametes produce the 2 homozygous forms and the heterozygous form in a 1- to -2- to -1 pattern. Because neither allele fully masks the other, the genotype ratio is also the phenotype ratio. The evidence from a laboratory cell line applies this principle in a concrete biological setting. Are you looking for HESI A2 exam actual questions? Visit our website pokerexams.com to access HESI A2 exam study guide with actual questions and answers. Scan the QR code or click the link in the description to get HESI A2 exam answer key today. Question 9. Genetically similar cells are maintained under standardized culture conditions. 2 cube-shaped cells in a laboratory cell line have side lengths of 2 units and 6 units. Which cell has the greater surface minus area minus to minus volume ratio and can exchange material more efficiently per unit volume? Option A. The ratio depends only on chromosome number. Option B. Both have the same ratio. Option C. The larger cell. Option D. The smaller cell. The correct answer is option D. The smaller cell. Surface area increases with the square of linear size, whereas volume increases with the cube. As a cell becomes larger, volume grows faster than exchange surface. Smaller cells therefore have more membrane area available relative to the amount of cytoplasm they must support. The evidence from a laboratory cell line applies this principle in a concrete biological setting. Question 10. Genetically similar cells are maintained under standardized culture conditions. During a laboratory cell line, population growth slows when prey abundance becomes scarce. How should this factor be classified? Option A, a limiting factor. Option B, a trophic pyramid. Option C, a genetic code. Option D, a taxonomic domain. The correct answer is option A, a limiting factor. A required resource can restrict survival, reproduction, or population growth when its supply is insufficient. The strongest restriction may change as environmental conditions change. Both density-dependent and density-independent influences can limit populations. The evidence from a laboratory cell line applies this principle in a concrete biological setting. Question 11. Genetically similar cells are maintained under standardized culture conditions. In a pp times pp cross performed during a laboratory cell line, which genotype ratio is expected? Option A, 1 pp 1 pp. Option B, 1 pp 2 pp 1 pp. Option C, all pp. Option D, 3 pp 1 pp. The correct answer is option B, 1 pp 2 pp 1 pp. Each parent produces 2 types of gametes in equal proportions. The 4 equally likely combinations include 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. Genotype ratios count allele combinations rather than visible traits. The evidence from a laboratory cell line applies this principle in a concrete biological setting. Question 12. Genetically similar cells are maintained under standardized culture conditions. A cell from a laboratory cell line contains an unusually large number of structures specialized for water storage and turgor support. Which activity is this cell most likely performing at a high rate? Option A, passive storage of hereditary information. Option B, formation of an external mineral shell. Option C, random chromosome loss. Option D, water storage and turgor support. The correct answer is option D, water storage and turgor support. A large fluid-filled compartment helps maintain internal pressure in plant cells. Abundance of a cellular component usually reflects the demand for its specialized activity. The observation therefore links cell structure with the physiological work being emphasized. The evidence from a laboratory cell line applies this principle in a concrete biological setting. Question 13. Genetically similar cells are maintained under standardized culture conditions. A trait in a laboratory cell line shows codominance. Which observation best demonstrates this inheritance pattern? Option A. The trait is determined entirely by temperature. Option B. The alleles blend into an intermediate product. Option C. A heterozygote displays both allele products distinctly. Option D. A heterozygote always resembles only the recessive homozygote. The correct answer is option C. A heterozygote displays both allele products distinctly. In codominant inheritance, both alleles are expressed rather than one masking the other. The phenotype contains recognizable contributions from each allele. An intermediate blended appearance instead describes incomplete dominance. The evidence from a laboratory cell line applies this principle in a concrete biological setting. Question 14. Genetically similar cells are maintained under standardized culture conditions. Oxygen becomes unavailable to cells in a laboratory cell line, but glycolysis continues for a limited time. Why can fermentation help maintain ADP production? Option A. It regenerates NAD+ so glycolysis can continue. Option B. It replaces glucose with DNA. Option C. It produces oxygen from carbon dioxide. Option D. It creates a new nucleus for each cell. The correct answer is option A. It regenerates NAD+ so glycolysis can continue. Glycolysis requires an oxidized electron carrier to accept electrons. When the electron transport chain cannot operate normally, fermentation transfers electrons to an organic molecule and restores the carrier. The pathway yields far less ADP than aerobic respiration but supports short-term glycolytic activity. The evidence from a laboratory cell line applies this principle in a concrete biological setting. Question 15. Genetically similar cells are maintained under standardized culture conditions. During a laboratory cell line, a researcher records that the mean stem length is 14.2 centimeters. How should this observation be classified? Option A. Qualitative data. Option B. Quantitative data. Option C. A hypothesis. Option D. A taxonomic rank. The correct answer is option B. Quantitative data. A numerical measurement with units is quantitative evidence. Such data can be summarized statistically and compared across groups. Descriptive categories without numerical values are qualitative. The evidence from a laboratory cell line applies this principle in a concrete biological setting. Question 16. Genetically similar cells are maintained under standardized culture conditions. In a laboratory cell line, a researcher selects only the healthiest organisms for the treatment group but selects organisms randomly for the control group. Which problem is introduced? Option A. Independent assortment. Option B. Taxonomic uncertainty. Option C. Selection bias. Option D. Osmotic pressure. The correct answer is option C. Selection bias. The groups differ before the treatment begins, so health status could influence the measured outcome. Random assignment or matched selection helps make groups comparable. A biased starting sample weakens the ability to attribute differences to the treatment. The evidence from a laboratory cell line applies this principle in a concrete biological setting. Question 17. Genetically similar cells are maintained under standardized culture conditions. During a laboratory cell line, an enzyme reacts with one substrate but not with another molecule of similar size. Which feature most directly explains this specificity? Option A. Every enzyme binds all molecules equally. Option B. The active site has a complementary shape and chemical environment. Option C. Substrates must be larger than the enzyme. Option D. The enzyme contains the organism's full genome. The correct answer is option B. The active site has a complementary shape and chemical environment. Binding depends on three-dimensional fit and compatible chemical interactions between a catalyst and its substrate. A similar sized molecule may still fail to align correctly or form the required bonds. Specificity allows cells to regulate many reactions independently. The evidence from a laboratory cell line applies this principle in a concrete biological setting. Question 18. Genetically similar cells are maintained under standardized culture conditions. A highly active cell in a laboratory cell line has many folds in its plasma membrane. Which advantage is most directly produced by these folds? Option A. More surface area for transport proteins and exchange. Option B. Less contact with the surrounding environment. Option C. Elimination of concentration gradients. Option D. A permanent decrease in membrane fluidity. The correct answer is option A. More surface area for transport proteins and exchange. Membrane folding increases available boundary area without requiring an equivalent increase in cell volume. Additional area permits more channels, carriers, receptors, and enzymes to be embedded. This structural adaptation supports faster exchange and specialized processing. The evidence from a laboratory cell line applies this principle in a concrete biological setting. Stressed about your HESI A2 exam? We can take HESI A2 exam for you remotely and undetected. We always guarantee a pass and payments only after results. To send us an email, scan the code or tap the link in the description and let's get it done. Question 19. Genetically similar cells are maintained under standardized culture conditions. Which process removes carbon dioxide from the atmosphere and incorporates its carbon into organic matter in a laboratory cell line? Option A, decomposition only. Option B, cellular respiration. Option C, photosynthesis. Option D, combustion. The correct answer is option C, photosynthesis. Autotrophs use carbon dioxide as a carbon source for building sugars and other organic molecules. Respiration, decomposition, and burning commonly return carbon dioxide to the atmosphere. The carbon cycle links these opposing transfers among organisms, air, water, and soil. The evidence from a laboratory cell line applies this principle in a concrete biological setting. Question 20. Genetically similar cells are maintained under standardized culture conditions. A cell in a laboratory cell line uses energy to move ions against a concentration gradient. Which molecule most directly provides readily usable energy for the membrane pump? Option A, carbon dioxide. Option B, ATP. Option C, cellulose. Option D, DNA. The correct answer is option B, ADP. Hydrolysis of a terminal phosphate bond can release energy and transfer a phosphate group to a protein. This coupling changes the shape or activity of transport machinery. Genetic information and structural polysaccharides do not serve as the cell's immediate energy currency. The evidence from a laboratory cell line applies this principle in a concrete biological setting. Question 21. Genetically similar cells are maintained under standardized culture conditions. A mature forest in a laboratory cell line is removed by fire, but soil and seeds remain. Which recovery pattern is expected? Option A, primary succession beginning without soil. Option B, no succession because fire prevents future life. Option C, secondary succession beginning with organisms that colonize the existing soil. Option D, immediate restoration of the original mature community. The correct answer is option C, secondary succession beginning with organisms that colonize the existing soil. When a disturbance leaves soil in place, recovery can begin from surviving seeds, roots, microorganisms, and nearby colonists. Grasses and fast-growing plants often establish before shrubs and later trees. Primary succession instead starts where soil has not yet formed. The evidence from a laboratory cell line applies this principle in a concrete biological setting. Question 22. Genetically similar cells are maintained under standardized culture conditions. Cells in a laboratory cell line carry out aerobic cellular respiration. Which overall change best describes the process? Option A, light energy is used to build glucose from carbon dioxide. Option B, energy in glucose is transferred to ATP while oxygen is reduced and carbon dioxide is released. Option C, oxygen is produced from glucose without electron transfer. Option D, ATP is converted entirely into DNA. The correct answer is option B. Energy in glucose is transferred to ATP while oxygen is reduced and carbon dioxide is released. A series of oxidation-reduction reactions removes high-energy electrons from organic fuel. Electron transport ultimately transfers them to oxygen, while released energy supports ADP formation. Carbon atoms from the fuel leave mainly as carbon dioxide. The evidence from a laboratory cell line applies this principle in a concrete biological setting. Question 23. Genetically similar cells are maintained under standardized culture conditions. Plants in a laboratory cell line cannot use atmospheric nitrogen gas directly. Which biological activity commonly converts nitrogen into forms that enter food webs? Option A, nitrogen fixation by certain microorganisms. Option B, diffusion of proteins into the atmosphere. Option C, photosynthesis by all animals. Option D, mitosis in plant roots. The correct answer is option A, nitrogen fixation by certain microorganisms. Specialized prokaryotes convert atmospheric nitrogen into ammonia or related compounds. Other microbes and plant processes transform these compounds into forms used to build amino acids and nucleotides. Consumers obtain nitrogen by eating organisms or organic matter. The evidence from a laboratory cell line applies this principle in a concrete biological setting. Question 24. Genetically similar cells are maintained under standardized culture conditions. Which sequence correctly traces information flow during protein production in cells from a laboratory cell line? Option A, ATP is translated directly into RNA. Option B, mRNA is replicated into a chromosome without enzymes. Option C, protein is transcribed into DNA, and DNA becomes lipid. Option D, DNA is transcribed into mRNA, and mRNA is translated into a polypeptide. The correct answer is option D, DNA is transcribed into mRNA, and mRNA is translated into a polypeptide. A gene first serves as a template for an RNA transcript. Ribosomes then interpret the transcript and join amino acids in the encoded order. The pathway transfers sequence information without converting protein back into genetic material. The evidence from a laboratory cell line applies this principle in a concrete biological setting. Question 25. Genetically similar cells are maintained under standardized culture conditions. One nucleotide is inserted near the beginning of a coding sequence in a laboratory cell line. Why can this mutation have a large effect on the protein? Option A. It shifts the reading frame for many downstream codons. Option B. It affects only the inserted nucleotide and no others. Option C. It prevents DNA from containing bases. Option D. It always changes DNA into lipid. The correct answer is option A. It shifts the reading frame for many downstream codons. Translation reads nucleotides in consecutive groups of 3. Adding a base changes how the downstream sequence is divided into codons unless the insertion is a multiple of 3. Many amino acids and possibly the stop position can therefore change. The evidence from a laboratory cell line applies this principle in a concrete biological setting.